Let's take a look at the photosynthesis gizmo in the Explore Learning website. We'll first launch our app, and then we have our gizmo setup. There are three variables that we can change here. We can change the temperature of the water with our aquatic plant. We can change the light intensity, how bright it is, and we can change the level of carbon dioxide that is present in the water available for the plant. We can see bubbles coming from the plant, and that will be the oxygen that is being generated by the plant through its process of photosynthesis. Notice that here there are 4 tabs, 3 of which are important to us: the table, the bar chart, and the graph. The most useful of which right now is the bar chart. Make sure that show numerical values is enabled. By changing the temperature, we can see on the bar chart how much oxygen is being produced. It's a nice visual comparison. Too much or too high of temperature will have a restriction on the level of oxygen produced, as well as too low of a temperature. There is a range where the oxygen level is constant at its maximum despite the changes in temperature. We can also change the light intensity to find what is the maximum rate of oxygen flow of production. We can see here it's about 48 milliliters per hour. Even though if we increase more light, there seems to be no effect. That means that some other factor is controlling the rate of oxygen production. That is our limiting factor. Similarly, we can increase or decrease the amount of carbon dioxide in the water to see its effect on the oxygen concentration. You can change these 3 things: temperature, light, and carbon dioxide to find what is the optimal condition to produce oxygen. Another series of experiments we can perform is that we change to the color tab now so that we can also change the color of the light available. And we look at the graph. Make sure that the x-axis, or manipulated variable, is set to wavelength, and the recording, the oxygen flow, that is our responding variable. We can keep these constant and change. We can change it to purple light, and then we can click on record data. It will record that at purple light, about 410 nanometers, we're producing about 32, 33 milliliters of oxygen per hour. We can change the wavelength to, let's say, click on record, and we get another data point. We can continue to do this, and we should be able to see a nice chart. Click on this camera icon to take a screenshot of your graph and include it in your assignment. This will be very useful for you to determine what is the best wavelength for oxygen production and the worst wavelength for oxygen production. Similarly, you can also look at the bar graph here to see the optimal rate of photosynthesis. There are a lot of other things that you can play around with in this simulation. There are 3 things you can change to measure the rate of oxygen production in this aquatic plant. If you have any questions, comments, please leave a comment below.